Hi, welcome back. So in the first section, we will be learning about the overview of PCR. So what is some of the important things about PCR? So what is PCR? PCR, Polymerase Chain Reaction, is a laboratory tool that is used to multiply copies of a particular fragment in the DNA. So it is a tool to multiply millions or even billions of this copy for certain uses. So for example, the DNA region by itself that we want to multiply can be used for detection purposes or medical diagnostics, biotechnology and medicine. Example, during this pandemic COVID-19 times, we know that this particular way of identifying whether a person has the COVID-19 disease is by taking the swab sample from a patient, particularly from the nasal swab, and this swab will be taken to the lab where the medical lab technologists or science officers will analyze the specimen. So the analysis itself is by using PCR, whereby the MLT will use PCR. MLT is medical lab technologists, so they will use PCR to multiply the DNA region of that COVID-19 virus. So the COVID-19 virus is called SARS-CoV-2. So if in that particular specimen there is the DNA for SARS-CoV-2, then it shows that the patient has the COVID-19 disease. And there are also other uses of it, for example in crime scene investigation, where we found some fragments of hair. So we will take this fragment of hair, do some analysis of PCR, and then identify and match with the DNA of that suspect and see whether the DNA from that hair contains the DNA of that particular suspect. So this is also uses the PCR method. So how do scientists analyze this? So one of the ways is by visualizing it through gel electrophoresis. So the aim of PCR to multiply at a sufficient amount so that we can detect that particular fragment of interest. So let's say if that particular fragment of interest is present. Now to visualize this is by using gel electrophoresis, this figure in black over here. So for example, you look at the lane where it says number 1. So number 1, for example, maybe suspect number 1, suspect number 2. Or let's look at the COVID-19 case. Specimen 1 is for patient 1 and 2 for patient 2. So let's say that the COVID-19 spike protein is of bandwidth 353 base pair. So that is the size of that particular DNA fragment. And patient 1, you can see that there is the fluorescence color that shows that the band is present. And number 2, the band at 353 is not present. So it shows that in the sample 2, there's no presence of spike protein DNA for COVID-19 virus. Hence, we can say that the patient number 2 is free of this virus. So this is how you visualize this amplified copies of that particular fragment. And then this is the use of PCR for this medical diagnostic purposes. You can see the figure down there. This is another use whereby this PCR copies, so for a particular region, once it has been multiplied, it can be used for cloning purposes.